Hi, my name is Ruchelle, and today I will be teaching you about the basics of heredity and the Punnett square. Heredity is the passing of traits from parents to offspring. One of the easiest ways to calculate the probability of inheriting a specific trait is by using a Punnett square. However, in order to successfully learn how to calculate the Punnett square, first I will teach you the basics of heredity. Such includes the Gormandel experiment, alleles, phenotype and genotype, and the law of dominance. There are many studies and ideas on heredity that evolved over the years. The following are only some of them. Aristotle proposed that male and female semen makes a conception. Antony van Leeuwenhoek said that there were animalcules in the sperm of men and animals. A lot of people who made theories had the right ideas, but no right experiment or evidence to prove their theory. Finally, there is Gregor Mendel, who is an Austrian monk. You may already know him since he is famous for conducting the pea plant experiment. He studied the patterns of inheritance by breeding pea plants together. Through the experiment, he discovered that each pea plant carried two characters or letters for each trait that it had. He called these characters alleles. Alleles are represented by two letters. An uppercase letter would represent the dominant allele, and a lowercase letter would represent the recessive allele. A set of alleles would contain two letters and will represent one trait. A set of alleles can either be homozygous or heterozygous. In the homozygous set of alleles, both letters are the same. Also, having a recessive homozygous set of alleles is the only way that a recessive trait can be shown. A recessive trait is a set of alleles in which both letters are lowercase or recessive. In the heterozygous set of alleles, one letter is uppercase and one letter is lowercase. Although the recessive allele is present, only the dominant trait will be shown, since the dominant allele always dominates the recessive allele. This is stated in the principle of dominance, where the presence of dominant allele prevents the expression of the recessive allele. Finally, we have genotype and phenotype. Genotype is represented by the set of alleles. As an example, we are going to state the genotype of a heterozygous tall pea plant. Recall that heterozygous means that there are two different letters. For pea plants, tall is a dominant trait and short is a recessive trait. So this means that the correct answer will be heterozygous TT. Next, we have phenotype. The appearance of an organism is its phenotype. You can find the phenotype of an organism by knowing its genotype. As an example, we will be stating the phenotype of a pea plant who has the genotype homozygous TT for height. Note how both T's in the genotype are lowercase, meaning that they are recessive. Also recall that a recessive trait can only be shown through a homozygous recessive set of alleles. In this case, the phenotype of the plant will be short pea plant. Take note and grasp onto the concepts that you have learned so far, since you will be needing them in order to learn about the Punnett square. It was invented by British mathematician and biologist Reginald Punnett. It shows the possible phenotype and genotypes of the offspring resulting from a cross between two parents. You are now going to learn how to interpret and solve a Punnett square. We will be using the cross between parent number 1 and parent number 2 as an example. Parent number 1 is homozygous for capital RR, and parent number 2 is homozygous for lowercase rr. The letter R will represent the trait or color. Capital R will stand for red, and lowercase r will stand for white. First off, the top and the left side are where the genotypes of the parents go. Parent number 1 will go on the top, parent number 2 will go on the side. The order of where the parents go doesn't really matter. Above or beside each column and row, there should be one allele from each parent. So we distribute the alleles from each parent accordingly. Parent number 1's set of alleles will go on the top, and parent number 2's set of alleles will go on the side. Notice how there's one allele stated above each column now. Write the allele down into the corresponding boxes below. For the first allele of parent number 1, we will list it down into the 2 boxes below it, like so. And we do the same for the other column. Notice how there is also one allele stated beside each row. Now write the allele into the corresponding boxes beside it. It is the same concept as what we did for parent number 1, but this time not that the boxes beside each other are rows, not columns. Write it down like so. You do the same for the other row as well. Now you have the genotypes of the offspring, and all of them are heterozygous RR. Next, you're going to learn how to formally state the genotype and phenotype. I've already given you the format for both the genotype and phenotype. All you have to do is insert the information within the brackets. Now think of the boxes in the Punnett square as 25%. Since all of the boxes contain the genotype heterozygous RR, the genotype will be 100% heterozygous RR. For the phenotype, you list a number of offspring that have the specific trait. Then beside that, you list the characteristic for which genotype is represented. Heterozygous RR stands for red. Therefore, the phenotype is for red. I will go over another example with you. This time it will be in the form of a word problem. The cat who is homozygous for small paws mates with another cat who is heterozygous for big paws. What is the genotype and phenotype of their offspring? You can choose to pause it here to see if you can do it by yourself, or keep playing to see the answer to the question. As you can see, I distributed the genotypes of the parents on the top and the left side of the square. I also distributed the alleles of each of the parents accordingly, like what I have taught you in the previous example. The genotype will be 50% heterozygous TT, since there are two heterozygous TT in the square, and each of the boxes represent 25%, and 50% homozygous TT, since there are also 2 homozygous recessive TTs in the square. The phenotype will be 2 big pod and 2 small pod. Hopefully you've now grasped the idea of this square, as we have come to the end of the video. It is now time to recap. A set of alleles can either be homozygous, 2 same letters, or heterozygous, 2 different letters. The principle of dominance. If a dominant allele is present, the recessive allele will not be expressed. The genotype is made up of a set of alleles. Phenotype is the appearance of an organism. Punnett square is useful for figuring out possible phenotypes and genotypes of the offspring. That is all, and I would like to thank you for watching my video, and I hope you found this video helpful.